Dear students, the topic is effect of hypotonic, isotonic and hypertonic solutions on red blood cells. Today we can explain osmosis by preparing isotonic, hypotonic and hypertonic solution. Put a drop of blood and observe under the microscope. Living cells have the potential to gain or lose water from the surrounding extracellular fluid through osmosis. The net movement of water into or out of the cell is driven by the differences in osmotic pressures between the extracellular fluid and the intracellular fluid. Thus, the effect that an extracellular solution has on the osmotic movement of water into or out of the cell is described by the tonicity of the extracellular fluid. If the osmotic concentrations of the intracellular fluid and the extracellular fluid are the same, then none of the solute can pass through the cell membrane. Then the osmotic pressures of the intracellular fluid and the extracellular fluid will be the same and no net osmosis will occur. The extracellular solution is said to be isotonic or with equal tension, referring to equal osmotic pressures. Cells removed from the body and placed in isotonic solution will retain the normal shape they have in the body. If a cell is placed into a solution with a higher osmotic concentration than the intracellular fluid, then the osmotic pressure of the extracellular fluid will exceed that of the intracellular fluid. As a result, water will flow out of the cell into the extracellular fluid, causing the cell to shrink and crenate. In this case, the extracellular fluid is said to be hypertonic, that is greater tension. Conversely, if a cell is placed in a solution with a lower osmotic concentration, for example, distilled water or like with a low concentration, then the osmotic pressure of the extracellular fluid is less than that of the intracellular fluid. As a result, water flow into the cell, causing it to swell, perhaps so much that the cell may undergo lysis or burst. In this situation, the extracellular fluid is said to be hypotonic. We can see the experiment with RBC. That means we are putting the RBC in hypotonic, isotonic and hypertonic solutions and see what happens. For this experiment, we prepared 0.9% sodium chloride solution as isotonic, 10% sodium chloride solution as hypertonic solution and 0.45% sodium chloride as hypotonic solution. Wipe out the tip of the index finger, prick with a needle, take 3 drops of blood in a slide, add 1 drop of hypotonic, isotonic, next hypertonic solution and keep it for 20 minutes. Observe under the microscope. When we observe under the microscope, in isotonic solution, the RBCs are normal. See, the size and shape of the RBCs are normal. But red blood cells in 10% sodium chloride solution, that is hypertonic solution, causes the cell to shrink and become deformed or crenated. See, in hypotonic solution, water moves into the blood cells, causing them to swell and become larger and more spherical. They rupture and lose all content and color, making them very difficult to see. These are called red blood cell ghost. From this experiment, we can understand that when a cell is put in hypertonic solution, what will happen? Shrink occurs. That is due to exosmosis. In isotonic, there is no change. That means the cell having normal size and shape. But in hypotonic solution, RBC will burst or lysis occurs due to endosmosis. Thank you for watching. See you in next class with another topic.